Picture a laboratory in Dallas, Texas. Rows of incubators hum quietly. Inside one of them sits an embryo that technically shouldn't exist. Its DNA is a patchwork, part living animal, part something that vanished from Earth thousands of years ago. Scientists aren't just studying extinct animals anymore. They're rebuilding their DNA letter by letter and putting it back into the world. And here's the part that should stop you cold. At least one of these projects has already worked. Not in theory, not in a simulation. A real animal, extinct for years, was born alive. We've spent the last few years watching biotech companies race towards something that used to belong entirely to science fiction. Today we're counting down 5 species currently caught in that race, from the one still years away from a single living cell, to the one case where extinction was, for a few minutes, actually undone. By the end of this list, you'll know exactly how close, or how far, science really is from reversing extinction. And number 1 already happened. You just might not have heard about it. Number 5. The passenger pigeon. September 1st, 1914. Cincinnati Zoo. A bird named Martha, alone in her cage, dies of old age. She is the last of her kind on Earth. Zookeepers had offered a cash reward to anyone who could find her a mate. Nobody ever did. That's almost impossible to square with what this species used to be. In the early 1800s, passenger pigeon flocks were so massive that witnesses described the sky going dark for hours, sometimes days, as a single flock passed overhead. Naturalist estimates put their numbers in the billions, possibly the single most abundant bird species that has ever existed on the planet. Imagine a flock so wide it would take a modern jetliner longer to fly past it than to fly from New York to Boston. That was the passenger pigeon. And then, in the span of a few decades, commercial hunting and deforestation erased it completely. So how do you rebuild a species from a bird that's been gone for over a century? You don't clone it. There's no viable cell left to clone. Therefore, the nonprofit Revive and Restore, under scientist Ben Novak, is taking a different route entirely. Since 2012, the project known as the Great Passenger Pigeon Comeback has been sequencing DNA pulled from preserved museum specimens, comparing it against the genome of the passenger pigeon's closest living relative, the band-tailed pigeon. Using CRISPR gene editing, the team is slowly rewriting the band-tailed pigeon's genome, one trait at a time. The longer tail, the reddish breast, the migratory instincts that once darkened American skies. Here's where it gets strange. This isn't resurrection in the way people imagine it. There is no passenger pigeon DNA sample intact enough to build a whole animal from scratch. What Novak's team is creating is closer to a genetic echo, a living bird carrying enough rewritten code to look, fly, and behave like the original, without technically being a clone of it at all. But that's exactly where the skepticism creeps in. Critics point out that a band-tailed pigeon with a handful of edited genes is still, fundamentally, a band-tailed pigeon. Even Revive and Restore doesn't claim otherwise. They describe the goal as restoring an ecological role, not resurrecting an identical organism. The team hopes to see the first hatchlings sometime between 2029 and 2032. Still years away, still speculative, still, undeniably, real work happening right now. So the passenger pigeon sits at the very edge of what's possible. A species being rebuilt from fragments and educated guesses. What happens when the animal you're chasing left almost nothing behind? You'll see that problem solved very differently in our next case, because this next species didn't just leave bones. It left something scientists never expected to find at all. Number 4. The thylacine, better known as the Tasmanian tiger. September 7, 1936, Hobart Zoo, Tasmania. A striped dog-like marsupial named Benjamin dies alone in his enclosure, locked out of his shelter overnight in the cold. He is the last confirmed thylacine on Earth. Less than 2 months earlier, the species had finally been granted legal protection. It came too late by weeks. The thylacine looked like nature's own experiment in mixing categories. Roughly the size of a coyote, with a dog-like head, a stiff kangaroo-like tail, and dark tiger stripes running across its lower back, it was Australia's only marsupial apex predator. Evolution converging on a wolf's body plan using completely different genetic material. Hunted for bounty, blamed for livestock deaths it likely didn't cause. It was wiped out therefore not by one single event, but by decades of systematic persecution. In 2022, Colossal Biosciences, the same company behind the mammoth project you'll see later on this list, launched a full thylacine de-extinction program. Their approach starts with the fat-tailed dunnart, a small carnivorous marsupial, and the thylacine's closest living relative. In October 2024, Colossal announced something remarkable: a reconstructed thylacine genome that was over 99.9% accurate, built using DNA extracted from a 110-year-old head, skinned and preserved in a jar of ethanol, forgotten for a century in a museum cupboard. Here's the twist. That same team then took things further than almost any de-extinction project before it. Using the preserved head, researchers recovered fragments of RNA, a molecule far more fragile than DNA, one that almost never survives this long. That RNA let them study what the thylacine's brain, tongue, and eyes were actually doing at a molecular level shortly before death. And in the lab, scientists have since edited more than 300 unique genetic changes into a single dunnart cell line, currently the most heavily edited animal cell ever produced. They've even built a prototype artificial uterus and used it to grow marsupial embryos more than halfway through a full pregnancy. But there's a hard limit to celebrating too early. No living thylacine embryo has been born. No Joey has taken a breath. What exists right now is a rewritten cell line and a genome on a hard drive. Genuinely extraordinary science, but still separated from an actual animal by a series of steps nobody has ever completed before. Some researchers connected to the project have floated a timeline of roughly 8 years before a thylacine could be reintroduced to the wild. Ambitious, unproven, but no longer impossible. A century after Benjamin died alone and unprotected, his species' genetic code is sitting in a Melbourne lab, more complete than anyone thought possible. Is that a resurrection in progress, or the most detailed eulogy ever written? Either way, the thylacine team isn't the only one working with a bird brain and borrowed DNA, because our next case belongs to the single most famous extinct animal on the planet. Number 3. The dodo. Mauritius, sometime in the late 1600s. A flightless bird roughly the size of a large turkey, picture something between a goose and a small ostrich, waddles through coastal forest it has called home for thousands of years, with no natural predators, and therefore no fear of the sailors, dogs, pigs, and rats that had just arrived on their ships. Within decades, it was gone. The exact date of the last sighting is debated, but by most estimates the dodo had vanished entirely by around 1681, roughly 400 years before this video was made. The dodo became something bigger than a single species. It became the cultural shorthand for extinction itself. Dead as a dodo entered the English language within a century of the bird's disappearance. So it's fitting, in a strange way, that it's also become one of the biotech industry's flagship de-extinction targets. Colossal Biosciences is running the dodo project using the Nicobar pigeon, a strikingly iridescent bird found across parts of Southeast Asia and the dodo's closest living relative. Yes, the dodo was, genetically speaking, an enormous pigeon that forgot how to fly. In September 2025, Colossal announced a genuine world first. They had successfully cultured primordial germ cells, the precursor cells that eventually become eggs and sperm, from pigeons for the first time in scientific history. The company has since established a breeding colony of Nicobar pigeons in Texas and begun the delicate work of editing those cells to carry dodo-specific traits. Here's the catch, and it's a strange one. Birds can't be cloned the way mammals can. There's no simple way to transfer a nucleus into a bird egg the way scientists did with the bucardo you're about to see at number 1. So instead, Colossal has gene-edited chickens to act as surrogate hosts, incubating germ cells that aren't their own. If it works, those chimeric chickens grow up carrying dodo-edited reproductive cells, and when 2 of them are bred together, their offspring could hatch out as something resembling a dodo, born from a chicken, genetically directed by a pigeon, standing in for a bird extinct for 4 centuries. Still, could hatch is doing a lot of work in that sentence. Comparative genomics between the dodo, the Nicobar pigeon, and extinct relatives like the Rodriguez solitaire is still ongoing. No dodo embryo has been created. No chick has pipped its shell. What Colossal has proven is that the pipeline, germ cells, chimeric surrogates, targeted edits, actually functions in pigeons. That's a real technical milestone. It is not, yet, a bird. 400 years after the dodo became a punchline for extinction itself, a colony of pigeons in Texas may be quietly rewriting that joke. But if you thought rebuilding a giant flightless pigeon was ambitious, wait until you see what the same company is doing with an animal that weighed as much as a school bus. Number 2. The woolly mammoth. Roughly 4,000 years ago, on a remote Arctic outpost called Wrangel Island, the very last woolly mammoths on Earth die out, a shrinking, inbred population, the final holdouts of a species that once dominated the Ice Age tundra from Spain to Canada. To put that in perspective, the pyramids of Giza had already been standing for centuries by the time the last mammoths disappeared. This wasn't some ancient, dinosaur-era extinction. It happened well within human civilization's timeline. An adult woolly mammoth stood roughly as tall as a school bus is long, weighing up to 8 tons, around the same as 4 full-grown African elephants. It carried 10 centimeters of insulating fat and multiple layers of shaggy hair built for a world locked in ice. It vanished, therefore, not from a single catastrophe, but from a mix of warming climates shrinking its habitat and human hunters closing in on the survivors. Colossal Biosciences, founded in 2021 by Harvard geneticist George Church and entrepreneur Ben Lamb, made the mammoth its flagship project, and arguably the most publicized de-extinction effort on the planet. Their approach, the Asian elephant, which shares roughly 99.6% of its DNA with the mammoth, edited at a few hundred key locations to restore mammoth traits. The shaggy coat, the cold-tolerant fat metabolism, the smaller ears. In March 2025, the company unveiled proof that the approach actually works, in the form of something almost comically small. Woolly mice. Ordinary lab mice engineered with 7 mammoth-inspired genes, born with the same long, wavy, gold-toned coat and accelerated fat metabolism the mammoth used to survive the Ice Age. Here's where it gets strange, and where the timeline suddenly compresses. In November 2025, Colossal acquired Viagen, the world's leading animal cloning company, responsible for cloning more than 3,000 animals across dozens of species. That single move folded decades of practical cloning expertise directly into the mammoth pipeline. As of early 2026, Colossal says it expects to produce its first mammoth-like elephant embryos this year, with the goal of live mammoth-like calves born by 2028, following the roughly 22-month gestation period of an Asian elephant. But, and this is the part scientists keep circling back to, even Colossal's own chief science officer, Beth Shapiro, has publicly acknowledged the limits of what's being built. When the company's related dire wolf project produced pups in 2025, Shapiro described them plainly as gray wolves carrying about 20 edits, not a resurrected dire wolf, and stated flatly that it's impossible to recreate an organism truly identical to one that used to be alive. Apply that same logic here, and a mammoth calf born to an Asian elephant surrogate is, technically, a heavily modified elephant wearing a mammoth's coat and metabolism, not a time machine. Still, walk that logic forward. If the animal looks like a mammoth, grows the coat of a mammoth, survives cold the way a mammoth did, and could restore the ecological role mammoths once played in the tundra, at what point does the label stop mattering? Whether the first calf arrives in 2028 or later, the tools built to get there, the cloning pipeline, the multiplexed gene editing, the emerging artificial womb technology, are already reshaping conservation science today. The mammoth gets the headlines, the embryos get the funding, but nothing on this list, not the pigeon, not the tiger-striped marsupial, not even the 8-ton Ice Age giant, has actually crossed the finish line. Only one animal on this entire list has. And it happened over 20 years ago, in a small lab in Spain, to an animal you've probably never heard of. Number 1. The bucardo, also known as the Pyrenean ibex. January 6, 2000. The Pyrenees Mountains, northern Spain. Rangers find the body of a 13-year-old female ibex named Celia, crushed beneath a fallen tree. She is the last bucardo left alive. With her death, an entire subspecies, one that had roamed these cliffs since the last Ice Age, officially goes extinct. The bucardo was a large wild goat, males reaching up to 220 pounds, roughly the weight of a heavyweight boxer, with thick, gently curving horns built for clashing on narrow mountain ledges. For thousands of years it clambered along the cliffs of the Pyrenees. But hunting had thinned its numbers for 2 centuries, and by the time conservationists tried to intervene, the population was already too small and too inbred to recover. Celia was the final one left standing. But here's the twist that separates this story from every other name on this list. A year before Celia died, wildlife veterinarian Alberto Fernandez Arias had captured her, taken a skin biopsy, and frozen living cells from her body in liquid nitrogen at -196 degrees Celsius. He didn't know it yet, but he had just preserved the only material that would ever let this species come back, even briefly. 3 years later, on July 30th, 2003, a team of Spanish and French scientists led by researcher José Folch attempted something no one had ever done before, cloning an extinct animal back into existence. They implanted nuclei from Celia's frozen cells into goat egg cells with their own DNA removed, then transferred the resulting embryos into surrogate mothers, a mix of domestic goats and ibex hybrids. It took 57 implanted embryos to get 7 confirmed pregnancies. 6 ended in miscarriage. Only 1 survived to term, and on that July morning a clone of Celia was born alive. For a few minutes the bucardo was no longer extinct. The newborn weighed about 2.5 kilograms, roughly the size of a house cat, and for a handful of minutes, this animal existed as the first and so far only species in history to be brought back from extinction and stand alive in the world again. Then her lungs failed. An autopsy revealed a malformed extra lobe in one lung and a condition called atelectasis, where parts of the lung fail to properly inflate. The clone lived somewhere between 7 and 10 minutes before dying of respiratory failure. Every other organ, researchers noted, looked completely normal. It was, specifically, her lungs that couldn't sustain what everything else in her body had already achieved. So is that success or failure? Here's the honest, uncomfortable answer researchers still argue about. It's both. The bucardo is the only animal on this list that has an actual, documented, undeniable answer to the question, can de-extinction work? The answer was briefly yes, and then no again, all within the space of 10 minutes. Scientists at the time weren't discouraged. Folch himself said the team wasn't especially disappointed, since deaths like this are common in cloning work generally, and that they intended to try again with better technique. Cloned cells from Celia are still preserved today, still being studied by researchers chasing the same goal that Colossal, Revive, and Restore. And every project on this list is chasing right now. For 10 minutes in the summer of 2003 in a lab in Zaragoza, an extinct species walked the earth again. Every mammoth embryo, every dodo germ cell, every rewritten pigeon gene on this list is, in some sense, still trying to catch up to what happened that morning, and to finally keep the lungs breathing past the 10th minute. So here's where that leaves us. 5 species. 5 companies and research teams racing along the same basic path. Recover the DNA. Find the closest living relative. Edit the genome. Grow the embryo. And hope biology cooperates. Some of them, like the passenger pigeon, are still years from a single living cell. Others, like the mammoth and the thylacine, are producing embryos and edited genomes at a pace that would have sounded like science fiction a decade ago. And one of them, the bucardo, already proved, however briefly, that the line between extinct and alive isn't as permanent as we once assumed. We started this video with a promise that de-extinction isn't hypothetical anymore. It's happening in labs right now, funded by billions of dollars, built on museum specimens and frozen cells and genomes read letter by letter. Whether any of these animals ever truly walks free in the wild again is still an open question, and maybe it should stay that way for a while longer. Because the real question this list leaves behind isn't whether science can bring these animals back. It's whether we've actually figured out what we'd be bringing back and what we owe the world once we do. If this kind of story pulls you in, you already know there's more of it out there. Stranger cases, closer calls, animals even further along than the ones on this list. Subscribe and we'll keep digging through the science that's rewriting what extinct actually means.